All right, let's get started then. I'd like to welcome you today to this EPI seminar, and I'm very pleased to introduce Katrina Armstrong. Katrina, who 20-some years ago was my student in the, it is true, my student, and who knew? No, we all knew that Katrina was a shooting star, but nobody knew how far she would go, and now, of course, she's gone, exceeded even our expectations, which were already high 20 years ago. And so Katrina is here to speak to us about unraveling mechanisms linking socioeconomic disadvantage. Katrina is the Jackson Professor at Harvard Medical School and the Physician-in-Chief in the Department of Medicine at Mass General Hospital, and she has many accomplishments, some of which we will hear today in her talk. So Katrina, welcome.
She's a member of our department.
Actually, and so my roots are, so for those of you who I don't know, I am an internist. I'm a general internist. I did all my training. And then in some kind of fortuitous streak of good luck, which actually at the time I saw as incredibly bad luck and blamed on my husband, I got uprooted from where I was training, which was at Hopkins for a very long time, and got moved to the University of Pennsylvania, which at that time I got put in an environment where I got a sense of the opportunity around. Is somebody taping me? Is that why this is on? Yeah. Okay. Or not on yet. Because usually people don't have trouble hearing me. I got offered an opportunity, and I actually will start with this very quickly, is I got offered an opportunity to join the clinical faculty in a clinical job or to be a fellow. And if you joined the clinical faculty, at that time you were going to work 8 sessions a week, and I was going to get paid $90,000 a year. And if I was joining a fellowship, I was going to get paid $50,000, I still remember this, $54,000 a year. But I asked them what I had to do, and they said nothing. And I was like, I can do that math, even without a statistics course. That means I'm getting paid infinity per hour. And I ended up doing a fellowship and a master's degree in clinical epidemiology and just fell in love really with this, which was the ability to use clinical data and to use clinical systems to try to understand health disparities, which have been my passion forever. And so Tim was a huge part of this for me, and it's just incredibly great that somehow I now get to see you even intermittently with years off my life in the traffic, even in Boston. So I am going to ignore that clicker. What I wanted to do today with you guys was really do something to talk a little bit about a journey for me. I'm going to talk about some research I actually did when I was a fellow in some ways under Tim and others. But then really talk about some recent projects that I think are what we call over at MGH, we have this saying we now call low rent epidemiology. Have you guys, I like this saying. And what basically it means is the ability to leverage existing clinical data systems that we have, stuff that we're collecting and using clinically, but to leverage it to answer what I think could be potentially important or interesting questions. I'm going to particularly focus on 2 examples, one of which is in press and one of which hopefully will be coming out soon, that focus around disparities-related questions, one in cardiovascular disease and one in cancer. But the overarching message of why I wanted to talk about this is I just think there's an increasing opportunity to think about how we leverage these systems to do things that are maybe not always going to require an R01, not always going to require what I would call high-rent epidemiology, not that we aren't all attached to those indirects. So I will say clinical systems, obviously the way we're doing them now, provide a window into the health and the healthcare of a population. We have a long history, right, of using those data systems or those types of the ways that we measure things to actually think about questions about the determinants of health in patient populations. Most of those systems have often been like a delivery system, like Partners being an example here, or a payer, like Medicare, right? Does that make sense to you guys? And I'm doing this in honor of you, Tim. I actually didn't care at all about prostate cancer until I met Tim. But then I was convinced that prostate cancer, I spent most of my time in breast cancer, actually endometrial cancer. Most of my initial grants were about disparities in endometrial cancer. But once you meet Tim, you do prostate cancer. And so in the beginning, I think when we thought about leveraging clinical systems in this area, a lot of we thought about it in disparities. We were really thinking about how do we take on this problem of prostate cancer mortality and incidence disparities between races in our country. These are just data that I'm sure you guys are aware of if you have Tim here, that show these persistent disparities in 2015 in incidence and mortality, particularly between black and whites in our country. The most common tradition of how we have used clinical systems, again, to take this on, I think has really been to study treatments, right? Things that are delivered in clinical systems. And so that is the tradition of how we've done this. Now, I've spent a lot of time in that area, and I'm just going to give you a couple of examples. So one is a study that we did very early on where we asked the question, why is it that there are large differences in rates of the treatment for early prostate cancer between black and white men? So we took one clinical system, at that time that was Penn Medicine, UPHS, and we discovered that when we went in and looked in those data, that 50% of white men in that system and 32% of black men with the same cancer, so early stage prostate cancer, got a prostatectomy. Now, at that time, we kind of thought prostatectomy was useful. It turns out maybe not, but let's just leave that over here for a second. And that we wrote a grant because we didn't understand why that difference existed. And that grant, we looked at a lot of different things from our clinical systems, including all of the comorbidity data, because one hypothesis was that reflected differences in comorbidities. We actually also, because you can get to patients through the clinical system, we surveyed all those patients, collected a huge amount of data on them, and looked at differences in their preferences for things about surgery. This is the study, and we had enrolled people. As you can see, there were substantial differences in in demographics between black and white men in this one clinical system looking at the prostate cancer. Turns out there were differences in comorbidities, but not as large as we had hypothesized. And there weren't specific differences in comorbidities as we had thought they might be. There were differences in race by preference for what the treatment was. So we measured how, using this system, all of the patients, what they preferred. Turns out nobody wants to die, just in case you guys think, I'm always amused that we go through this with I'm on service right now, which is adding to the complexity of my life. And one of the things we always get through is goals of care discussions. And it's an interesting moment because in essence, nobody really doesn't want to live as long as possible. It's like one of those things with the house staff. But if you looked at differences around concerns about incontinence, impotence, anesthesia, things that might be associated with surgery, there were definitely racial differences. But the reality was, and this was actually a great story, any of you guys research assistants in here? Anybody at that level? Or has anybody been a research assistant? No? I certainly have. So this is a great story, I think, about the power of having great research assistants. So we were I was sweating because all the models that we ran, we could measure all these racial differences, but they didn't predict treatment. Does that make sense? So the dependent variable over here, right, is whether or not people are getting prostatectomy. We can measure all these differences, and it's not telling us, it doesn't explain any of the racial difference. And the research assistant actually said, but you know what? Your patients are completely segregated in this system. We're enrolling patients at actually 3 hospitals, you remember, but I'm simplifying it for the point of the talk. At the time, HUP, the VA, and actually Presby. And it turns out that when you actually looked at it, the difference in treatment was just completely driven by where the person went for their care, right? So if you went to HUP, and this is actually true in medicine now too, you got paid a lot for a prostatectomy, and so people were much, much more likely to get prostatectomies. If you went to the VA, that wasn't how people were reimbursed, and the rates of prostatectomy were just much lower. And so it turns out that we were also highly racially segregated, so that many more black men, the proportion, 86% of black men were seen at the VA, where 28% of men underwent prostatectomy, and 70% of white men were seen at HUP, where 58% underwent prostatectomy. And so that is a classic kind of ability to use a clinical system to just answer a simple question about why we saw a disparity. That then led us to decide we were going to use other clinical data sources to try to look into this. And so we went from beyond a single patient health system cohort. Do people ask questions in this or not? You guys are welcome to interrupt me. Is that okay? I know we have to get kicked out at some point, which is fine. I promise I can cut this off wherever. But please, if you have questions, there'll be more recent stuff that you guys should ask about in a second. So in that, I think there's been a ton of work that uses national or large-scale claims data, right? So administrative medicine, to do basic epidemiology to understand the causes of disparities. And so often those focus on treatment, but for us, this was actually an opportunity to try to understand how much segregation, so a variable at the community level, could be contributing to this problem. So what we did was we knew that there's a data that hospital level segregation was becoming increasingly recognized about this time as a major part of the experience of patients in our country. And our question was, this hospital segregation is largely driven in part by residential racial segregation. Is racial residential segregation associated with racial differences in treatment? So we knew within our one clinical system, if that makes sense, that we'd observe this effect. And then the question was not just does it happen at other hospital systems, but is it driven by the community level of segregation as that happen? So I'm not going to take you through all of this. It's really just an example to get to where I think we are now. So we did a study. Now the numbers are much bigger, right? Because we've got everybody within actually this is within the U.S. in a certain age time period that was diagnosed with early stage prostate cancer. And we're looking at what happened to them in terms of their treatment. Again, as you can see, large differences in the groups between blacks and whites. And I think as you go down the bottom, you can see that there are differences in the isolation index, so the level of segregation in the cities that they were in. This is not surprising, as we know about the distribution of racial groups in our country. And there are differences in the other characteristics about the MSAs that they were in. And so this is just one of the final slides from that project. I'm really just giving you this because it's an example, I think, of how people have traditionally used clinical data systems and clinical data to ask and answer questions related to clinical problems, and in my case, disparities. I will say this was also an example from my experience of why splines are not your friend. So I don't know how many of you guys spend your time with splines, but I think splines are the hardest thing to explain to anybody when I try to talk to about this. And it turns out that when we looked at the data, and this is now, I always forget, I still can't do left or right. I don't know if it's like a problem with my brain. But as you look on the left, these are prostatectomy rates versus the level of segregation down at the bottom in your MSA. And then these are on the right is radiation versus segregation. And the bottom line is it's complicated, right? It turns out actually that as the cities get more segregated, that everybody is less likely to get active treatment, which is an interesting issue with how we think about our cities and the level of healthcare that they provide. It does turn out that the level of disparity probably doesn't vary linearly with the level of segregation, which is what we had assumed might happen. Our hypothesis was that we might see that the disparity would get larger as segregation goes up. And so these were early studies that we did. I can't even remember. I was probably an assistant professor or something. It was a long time ago that really has been part of my passion of how do we leverage clinical data that we're collecting for other reasons to try to understand disparities in outcomes. So I now think we're at a new time where we have a much greater opportunity to do this in a way, in this kind of low-rent epidemiology way that can help us take advantage of these systems to really answer important questions. And that's because a couple of different things I'm just going to highlight here. So the first is, like, we collect one bucket of data in clinical care these days, way more than we ever used to. We're going to ignore that. No, just ignore it. Just ignore it. I'm sure it'll be fine. It won't break the computer at all. I'm not all there. I think it's okay. I really do. Tim and I go way back. Tim's office was always really neat. Have you guys noticed that? Have you guys seen, other than all the the name tags, do you still save your name tags or not? Yeah. Did you get okay. So wait, you're jumping ahead though now.
I
You're
am not doing
killing
anything.
me.
Yeah,
You turned it upside
the
down.
water is doing this.
I think it's fine.
Yeah, you think?
Yeah.
Okay,
Okay, but we're going backwards here.
if you say so.
I do.
All right.
Okay. So I think if you think about it, we collect so much more data. And I don't know how much you guys are aware of that, but in clinical medicine, in our health systems now, there's an enormous amount more data that is collected than there ever was before. And so there's almost nothing that we aren't interested in that we don't have some way of thinking about getting from the existing data collections. It's also true that stuff that we used to write really sexy parts in our grants about how we were going to use natural language processing to identify X and Y and Z from our notes is kind of standard practice in medicine right now. So we are using NLP all the time, just in clinical medicine, to try to pull out different variables for quality or for other stuff. It's become kind of standard in how we think about our systems. I think we're increasingly collecting biological data for clinical decision making. So stuff that we used to have to add on, like we're collecting in biobanks, we're going to collect this. We're using that in clinical medicine. We're actually in a whole program now where we're doing immunological profiling around certain types of patients that we used to think of as a huge add-on. It's also true, as you guys are aware, that there's a move to think about us being responsible for populations, right? So it's not just people who show up. It's actually the concept that there is I am accountable as a system for a large population of patients, which means the issue of missing data and loss and censoring in every direction goes way down, if that makes sense, because we understand that we have to find those people and figure out what happened to them. And then I will just say I think there's a much greater sense of a focus on variation, and I'm not going to talk a lot about that. So I want to talk about two examples that people that I've worked with have been working on. One, a woman named Anne-Marie McCarthy, who actually also was at Penn before, and then one cardiologist named Ahmed Tawakal that I've been working on to leverage data to answer questions I think are potentially relevant. So the first has to do with an opportunity to leverage how we do genotyping, so essentially something called snapshot that we do on tumors for women who have metastatic breast cancer. So this has become standard clinical practice for metastatic, and in some cancers for all patients, but for us for metastatic breast cancer patients. And so we have the ability to leverage those data to look at some questions. So something I've long been interested in before Tim got me into prostate cancer was disparities in breast cancer mortality. And it turns out, as we know, African American women are much more likely to die of breast cancer than white women. This is actually a paper that Anne-Marie published in the American Journal of Public Health some time ago with me. And basically, I didn't label it well, but up in the top left of your screen is the breast cancer mortality data over time for black and white women. And the paper basically makes the point that that disparity between black and white women hasn't changed over time really at all, even though we've seen some changes in other diseases that I won't go through here. But that we have now an opportunity to think about bringing together our clinical data systems, this information that we're collecting in these different types of biological assessments, to link clinical risk factor data with tumor data and with these types of outcomes. So one thing that is an example of that came from some time ago actually at Penn was when we first started collecting biobank data. So we first had the opportunity to go in and look at genotypes, so germline genotype in this area, and asked, does having that data help us anyway? This is a paper that Anne-Marie did where we basically asked the question, does having genotype information do we do better at predicting breast cancer amongst black women? Because there's been a long-standing challenge that a lot of the breast cancer risk prediction models don't work very well in certain subgroups. And so I'm not going to dwell on the specifics here very much, but if you look at it, so we looked at about 450 white women, and in this particular paper, there's a lot more since then, about 300 African American women. And essentially what we ended up finding was that we reclassified, if we included the genomic information, we reclassified a much greater proportion of black women as being high risk than we did for the white women. That it seemed like we were finding more risk alleles that were shifting us over a threshold in black women at the time than we did for this particular subset of allele, or subset of SNPs that were looked at in this time point for white women. And so it was an opportunity to leverage data we have just to get a hint and that we might be able to use these data to do better in risk prediction. There's been a lot more work done in this since that time. So one of the more exciting areas, though, I think, is really the understanding of the molecular changes in tumors, right? So the tumors, as we think about cancer, are really this unbelievably complicated tapestry of molecular changes that are driving cancer. And we know now, if we look at breast cancer, and this is just some data from the Cancer Genome Atlas, I'm not going to go through them, you guys can't see them, even if I could, but know that we have a range of mutations that we find when we look at tumors, and that information is now being used clinically. So it used to be that we were really just doing that in a way that we would have to add it on for research projects, but we collect these data in cancer patients routinely, and particularly in certain situations here in metastatic breast cancer patients. And so if you take what happens at Mass General, we're running for metastatic breast cancer patients essentially a large profile of the genomic signatures in those tumors, allowing us potentially to ask in some questions. So one of the questions that has come up is related to racial differences in a particular pathway, here PIK or PI3K or PIK3CA mutations in that pathway. And this is just data that actually came up from one of our residents, published this paper, Tanya Keenan, who was actually a Penn medical student, which looked actually in the Cancer Genome Atlas and showed differences in the rates of mutations by African American and white women in that. And I think what you can see is that if you look at it, there's differences in several of these, but particularly in PIK3CA mutations. So we were interested in asking, fundamentally, we don't understand right now, and the part of the story that gets a little bit complicated, is we know that those mutations lead to increased PI3K activity. So if you look at mutations in cellular models, they increase activity of what is we think to be a cancer, to promote cancer growth. So the pathway actually leads to multiple cellular processes that influence metabolism, motility, but it's crazy complicated. And because it's so complicated, it turns out that there's a lot of conflicting data about how this pathway may influence cancer outcomes. So if you take the basic epi studies that have been done, and probably people in here know these data a lot better than I do, but they've been shown multiple different relationships where either mutations appear to be protective in terms of outcomes, so people do better with PI3K mutations, or people do worse.
Are these all the same mutations?
Well, so most of them are in this helical and kinase domains, and so if they're in the helical and kinase domains, it hasn't appeared to be a mutation specific within those domains. Most of the mutations are in the domains that have been studied. There could be some mutation variation in this, but people just don't understand it right now. So Anne-Marie decided we could leverage our clinical systems to understand maybe they're modifiers of what's going on within these patients. So that if somebody has a mutation but is exposed here, she's going to end up studying aspirin use in this group. We study because we have great data on that and because of some obvious hypotheses I'll show here about how that might work, that maybe some of the reason that there's so much variation in outcome is partly because patients have different experiences, right? So we've got tumor data, but if we can bring in the clinical data about what's going on for those patients, maybe we can try to understand what's going on in that area. And so this is just a schematic that looks at this particular complexity of these pathways and basically adds the point that we also know that PI3K has effects on the COX and COX-2 pathways. And so that there's data in colon cancer, as many of you guys may be aware, that there's effects that make aspirin use may actually lead to improvements because of effects in these pathways. And so I'll just show you the data that we got here looking at about 300 women with metastatic breast cancer where we had all their tumor analysis. We had about 36% of these women had mutations, so a reasonable proportion of women with mutations in this pathway. And interestingly, and you guys are aware of this, but a lot of the risk factors have reasonable prevalence as we look at when we start talking about medication use in large clinical populations. So about 20% of women in these populations use aspirin. And so creating a kind of interesting natural experiment. They're not using it because of their breast cancer, let's be clear, but and not being able to understand this. Now I want to be very clear, we're just looking in this sample, we're taking advantage, this is the low rent epidemiology part, right? We're taking advantage of people who had their tumor sequenced. So we know what their tumors are. Everybody got their tumor sequence who came in. And basically what she finds, I know, what we find is that there's actually there's small effects in the overall population, not a big effect of what happens. You can see there's a slight improvement in survival if you do have a mutation in this particular gene in PIK3CA, which is what's been seen in some other studies also. So there's some improvements. Aspirin alone doesn't seem to make much of an effect. But interestingly, there is a big interaction, and the interaction stays no matter essentially how you test it. Where interestingly, there's a particular benefit. So the people who do best are the people who take aspirin and have a mutation in the pathway, which is not exactly what was seen in other studies or in other types of cancer. And so that holds also, just to be clear, if we look at survival. So we're looking both at time to metastasis. I'm sorry, I should have made that clear. This is time to metastasis in this analysis. And then this is survival. Same interaction effect where the aspirin and this mutation have a very different effect than one or the other alone. It also turns out these are just a regression where we're just looking at 5-year survival, just trying to understand if we were to categorize women, do we see the same thing? And I just put it up here because I think you can see the size of the difference down the bottom, that if you take an aspirin non-user, that having a mutation in this gene seems to be very protective, whereas if you take an aspirin user, it is less protective and potentially even associated with a worse outcome. And it's an interesting question. We don't understand. This is hypothesis in some ways adding. We understand the pathways. We understand what's going on in terms of some of the pathways, but we don't understand how that plays out in clinical populations. So we have this opportunity in some ways to take the biology and to understand these hypotheses and to bring them to our clinical data to test things in what I'm calling this low rent way, where we can understand if there's signals that we should be following up on. So I'm going to talk about another example that I think is really fun because it takes advantage of something that I've had a lot of interest in, which is trying to leverage the enormous amount of imaging that we do in clinical medicine for really honestly very little good from my perspective. I'm on the service right now, and essentially every patient gets admitted to the MGH, I swear, gets at least 3 parts of their body imaged for some reason. And so, imaging is very expensive. If you try to do imaging in a study, it's very hard to get imaging done. People have to go in and get that image. It's very cumbersome. It can be very tricky to get those types of data to add them on to understanding a clinical problem. So one of the problems, again, that I've been interested in is disparities. This one now is about cardiovascular disparities, although interestingly, we're doing the same types of studies in cancer and finding very, very similar things as I'm going to show you here. So we've known for a long time that socioeconomic status is associated with mortality from coronary disease in many different ways. It's often been studied using educational level. It's obviously the Whitehall studies. We know all of those types of studies. These are data just from the US and from Scotland On the left is Scotland, looking at socioeconomic status using a multi-level metric that actually stratifies. I've just shown you for women, but the data look the same for men in terms of the level of coronary mortality by the level of deprivation. And you can see the really just striking differences between those lines. And the United States, these are a recent study, or relatively recent, looks at which outcomes have the greatest association with county median income, and I'm going to come back to that because we're using area level for some of these analyses, and you can just see that heart disease up there is near homicide and having a fairly strong correlation with the socioeconomic status of the county of your surrounding area. It also, and that was the point of this paper, has a fairly strong correlation with income inequality, which is here on the y-axis. But I'm going to focus not for the moment on inequality, but just on socioeconomic status. So we decided to see whether or not we could look at biological pathways that were related to stress and actually to amygdala activation. So is there a way that we could use existing scanning, PET scans, that are going on in our clinical populations for, let's say, questionable indications some of the time, and look at whether or not by reading other parts of that PET scan, right? So when I order, let's say, a PET scan for cancer staging, in general, they're going to focus on certain things that they read clinically. But those images are stored, and I can pull them back out and reread them for other reasons to use them to study other parts. Here, studying the amygdala and studying the brain in scans that were done in people for other reasons. Does that make sense to everybody? And then we can also measure other biomarkers. Here, Ahmed Tawakol is particularly interested in arterial inflammation. So his interest is in whether this is all related to some inflammatory pathway that gets triggered by a stress response. So this is the patients who we put into this particular study. So if we looked at it, there were about 6,000 patients who underwent a PET-CT at MGH in this period, and then we excluded patients, as you can see, who had a chronic inflammatory condition, who had active cancer as the first exclusion, who had prior cardiovascular disease. You can look through those different things. And we ended up only with about 580 patients. But you can imagine actually to get full PET scans and full clinical data and all of these things on 580 patients in what I'm calling the low-rent epidemiology way is actually a significant number of people. We knew they were free of coronary disease. They had been extensively phenotyped. So we knew they were disease-free at entry. They'd had a PET scan for some other reason. And then we went back and reread those data. And so this is not my stuff, so I just want to be very clear. There are other people that do this. But what they basically do is go back and reread the PET scans to measure amygdala activity, categorizing it, and obviously it's a continuous variable, but here showing you a scan that has lower activity and a scan that has higher activity. You guys can see the little arrows. They help me out when I'm trying to figure out where it is. Hey, Jason. I think you're actually on this paper. And then looking at arterial inflammation in the same study in terms of measuring it. So this is the cohort. So there's about 509, or there are 509 patients in the cohort. I won't run through it, but one of the advantages of using clinical systems is that we have a ton of data on these individuals, and they get repeatedly measured, right? So if we have missing data, somebody is going to go back and find it for a lot of these individuals. The time series stamping is a big challenge when you do this. So making sure that we know when the measurement was actually ends up being one of our biggest problems. And because as you guys know, the way these things are entered in, well, as you may not know, but as I probably can tell you, sometimes things are entered in lumping dates together more than splitting days together. So if you need to know the exact day, I'll give you an example. When we try to look at labs, it turns out the date the lab is resulted, the date it comes back, is actually the date, it shows up as the date it was ordered sometimes. And so that doesn't always make sense, and then sometimes it shows up the other way. So understanding the dates can be really critical, but this was less about dates. So if you look at it, this all makes sense, right? So what Ahmed's done is divide the cohort into patients who ended up with an adverse cardiac event. So the MACE cohort, there's only 40 individuals over the follow-up here who did, and then the 469 who didn't. But all the risk factors track in the way that you would expect, right? So it suggests that we're finding that these individuals are much more likely to have the traditional risk factors. I will say I went home and showed my kids this current smoker thing, because it still just blows my mind, right? I mean, it's just an amazingly strong risk factor for everything. I don't think they were particularly impressed, but so if we look through this, there were also pretty striking differences in household income and in the rates of crime per zip code. This is using zip code measures we have for patients in terms of the differences in patients who had either no cardiac event in the middle column or a cardiac event in the right. This just says kind of the same thing, but this looks at survival without a cardiac event. Here now looking by the level of neighborhood income. And so it's really, I mean, it's still an extraordinarily striking effect that we see. So if you look at it, that the patients who are in the top income quartile and where they live have a much, much better survival without, are not getting cardiac events. And this is just within the 500. But as I showed you before, we've seen that in studies again and again across different places. But this is really what I wanted to show you, that they were able to do with this because of what the data were available. So then able to correlate the local income, so income in the local area, here zip code, with amygdala activity, so this sign of stress that we can pick up on the PET scan, and with arterial inflammation, another measure that they're getting from the PET scans. And I think as you can see, there's really a fairly striking correlation in our patients. These are people who are coming in and being seen, getting PET scans in our system. And this is in Boston, right, between the quartile of their zip code income and the level of stress that we pick up in their amygdala, completely unrelated to why they came in. Same if we look at arterial inflammation, so the one up on your top right. And then the panels on the bottom are not using income, but are using crime rate for the zip code. And so something we're getting from these data, and I'm not going to walk through this complicated kind of mediation pathway very much, but I want to just make the point that what we then did, and what Ahmed did, was go in and look and ask how much of these things then explain the overall relationship between socioeconomic status and adverse cardiac outcomes. And I think what you can appreciate is that there is something there, right? So that we're basically finding, using this single cohort from a clinical system, that socioeconomic status leads to, that your neighborhood socioeconomic status has a fairly strong relationship with the measure of stress that we can pick up by measuring amygdala activity. That then has a relationship to bone marrow activity, which has a relationship to arterial inflammation. And obviously these things are interrelated, but there's a pathway that explains a significant proportion of the socioeconomic status to the adverse cardiac outcomes. And I think the part that, obviously I think there's biology here that's interesting. Ahmed is particularly interested now in figuring out, we have patients who defy this, right? So if we take some of our folks, they have every adverse risk factor, both on the zip code level, but on other levels, but they seem to be completely stress-free. So if we look at their PET scans and we look at their inflammation, they are, as what we could call this kind of positive deviance, right? So despite the external risk factors, they are not experiencing this biological response. And I think he's particularly interested in linking this to other data we have, either from thinking about germline DNA, genotypes, we have other types of data to try to understand that as well. Okay, Tim, I'm so sorry. I'm really sorry.
taking it from you.
I know, but I really thought I was going to cough. I really thought I was going to cough. Okay, well, here we got more. This is reassuring all you guys that I'm on service, I'm sure. So I think the idea is that we can not only use these data to kind of create hypotheses to take advantage of the clinical data that we're collecting, but we can use them to identify people that we may want to do a deeper dive in, right? And then to try to understand, can we go from this to actually doing deeper studies where we're going to collect more data, larger cohorts, and bring in other samples. And I think the idea here now is probably to use this to identify a group that seems to be particularly resilient in this situation and see if we can understand the both biological factors that might explain that, potentially the clinical factors that explain that, and then the social factors that may be driving that also. And so, you know, I really just wanted to bring those examples up and then to take some questions here, because I think we've traditionally thought often, when I grew up doing epidemiology, that we've often used clinical data systems, I think, to think about answering questions about clinical stuff, right? So I grew up using clinical data systems to answer questions a lot about treatment, about other different types of things in the healthcare system. And I think we're at a time that, particularly for students and for junior faculty, I think there's an opportunity to think about how to leverage them to do what I do think is this kind of low-rent epidemiology side of it, where you can get access to data that may give you more examples that aren't necessarily going to be the definitive answer, does that make sense, but can leverage you then to go in and start asking questions in other samples or in other populations where those data may be much, much, much more expensive to collect, right? And so we're collecting these data now, and I think there's an incredible opportunity to access them and use them. And so I'm here kind of as an advertisement for that, if that makes sense. I think these are two examples that I've been involved in. Honestly, I probably could have pulled hundreds of stuff that is going on, not just across the system, but across lots of other places to think about it. I do think there's a lot more data there than we may be recognizing in different ways, and the ability to work with epidemiology and the clinical side to identify populations that are clean enough that we can actually think about where a question has internal validity. You're recognizing that the external validity, I think, will often require larger, different studies. But to be able to create those cohorts, I think we're at the point where we can do an incredible amount together with that. And so partly I just wanted to come and tell those stories. And despite being a little late, I'm actually finishing on time, so I'm very impressed with that. Okay, so I'd be delighted to take questions, and I apologize that I'm not all here, but anyway. Yeah.
Hi. So I'm a sociologist by training.
Oh, fantastic.
Your work on the SDS is very interesting. But I have a question about the last paper where
Yes.
you talked about with the imaging and whether or not you'd be able to isolate the both individual SDS or the contextual SDS.
Yes, so absolutely. And I mean, part of the reason I'm here is, you know, and I think, and I don't know, I mean, Jason, I don't know, but I think the ability to try to figure out how to do a better dialogue about what we're collecting in the health systems, how to also get your guys' input and what you want collected. Does that make sense a bit? So I'll give you an example. So we are now, because of our ACO, and Jason can talk more about this, we are collecting social determinants of health. Are you aware of this or not?
I know it's social.
No. Do you know what items we're using or not?
I mean, they vary from.
Well, so that's the rub. Let me just be clear. So I had a huge tantrum. Were you around for that about 2 years ago? I have many tantrums, but I had so we were all going to collect different things in our EMRs across Boston to try to measure characteristics that we might want, whether it's housing instability, food insecurity, like whatever you want. And then if you were to think about leveraging those data for what you guys might want to do with them, we're just like shooting ourselves in the foot, right? And so the two institutions that have led in that, and you may work at one, I don't want to is actually Boston Children's and BMC. And so they have led in figuring out the items we should use to actually put in the electronic medical record to collect. And so we pulled together a collaborative. I mostly had the tantrum, to be clear, but across a lot of the hospitals to try to argue that if we were going into this together, that we would collect the same items, right? Because right now, as I'm sure you're aware, we don't really ask people those things in healthcare, right? Which is why we've studied race forever, which is because we actually don't ask race either. We just guess it, but we put it in our systems. Does that make sense? So I would argue, and do you have a sense of when we will, over the next 3 years, across a large proportion of the populations who we are responsible for, particularly in the low SES populations, but increasingly across all, have a set of about 7 items. Have you seen them?
I've seen some.
Collected. And we should be able to get much better to, like, is this a neighborhood problem? Is this, I mean, my answer to your substantive question is it's both. Like, we know that these things are probably synergistic in different ways, and that is probably both an individual problem, we're seeing some effect of an individual level, and there's some neighborhood effect, would be my guess. Things like crime rate, some of these things are going to naturally be a small area or another types of level variable. But we don't have individual income in our data right now. No, is probably the bottom line. So we have to bring those things forward, but they're getting collected. And we need to collect them in a way that is useful for people. Does that make sense? Yeah.
Yeah, so I wanted to ask a question about, for students and
Yes.
early career faculty who might be interested in collaborating but are not physicians, how would you recommend that they go about, working with a clinical group within these kind of really unique data to address different hypotheses?
Well, and so I'll make a couple of suggestions there, and I'll make a couple of suggestions, but I'm here partly to have that dialogue. Does that make sense? And I don't want to sound like I have I don't know how to put it, like I have an easy menu exactly. And I do think it depends a lot on people, I've also discovered over the years. I mean, when Andy and I worked together in Penn, we spent a lot of time back and forth between the clinical problems and the clinical systems. And, I'll tell some stories. I used to have another tantrum, Andrew probably remembers, which is that we had these big meetings in Annenberg. We were doing a lot of stuff about cancer communication. And the agenda would be like the Annenberg scientists, and then it would be like the clinical input from Katrina at the bottom. Do you remember that? You don't remember that? Yeah. And I would be like, Bob, like, what are you doing? So I think there is a part of like mutual respect on both sides a little bit. Do you know what I mean? And so recognizing that a lot of the clinical faculty want to ask and answer these questions. They just don't know how. Does that make sense? So they don't necessarily want to be seen just as like the avenue to the data. They are desperately interested in having interesting conversations about what's happening in clinical areas. And so one of the things that we did before was try to set up groups that were interested in areas. Let's say we pick cardiovascular disparities. My latest obsession is what I'm calling the biology of inequality. We pick areas that groups are interested in and then figure out that common language. And then I think you have to throw, and I'd be happy to contribute to that, I think you have to throw enough little bit of pilot funding into it. Because one of the things I've learned in academics is that people will do anything for $5,000. It's like, it's an amazing thing. No, you like put out an RFA and say like, I'm going to give out pilot grants between $5,000 and $20,000, and like people like, it's amazing how people will come together. So I would suggest, and it doesn't have to be with this department in particular, but I would suggest if we were to find some shared priority areas that matter to the students, matter to the junior faculty, we were to link in the clinical side. I think mental health has done this a lot. I just want to say there's great examples, but I don't know that we've done it across all of these other examples.
But there are some people, both faculty members and FD, who are doing this, and also students
Yes.
who are. And Derek and I are both interested in this. We're all happy to be linked to this. There's a lot going on, and I would give a plug for on the EPI side is absolutely want to work with the clinicians because they understand why the data are there or missing in a way that you won't if you're not a clinician.
Right.
They'll understand about the error pattern, et cetera. But on the other side, clinicians and EPI people should work together on this because otherwise people like Google are doing it and they understand neither.
Right.
And so this is your chance to really contribute to the quality of the science.
But also I will say,
learn how to do it, and it's going to give.
and I will just say, Deborah, the other thing, being responsible for, I don't know how many, I try not to count, but I bet 800-900 doctors. The thing that they love the most is getting to work with somebody on a project, honestly. And so just in terms of you guys could single-handedly address doctor burnout. Think about that. I don't mean it quite, but I think people really want to work with people to answer these questions. So it's both they understand the data, but I think it's also something that those collaborations can be really fruitful long-term, I think, in terms of figuring that out. You do have to forgive people's flaws, right, on both sides and be willing to understand that people bring different things to it. So I do think mental health's been a great example, trying to have more people who cross back and forth and are able to think about that. But I think setting up those types of collaboratives, and then it's amazing what people will do for $5,000 in my experience, has been a huge thing. We have to leave, I think, right?
Let's thank you.
Thank you.
So happy to come.